Okay, thank you very much, Bill, and thank you also to the speakers in this session and also in the first session. We'll now engage in questions, and so I'll read through some of the questions that will occur. And Laura, how do I find questions that may come up for any of the speakers and not just for myself?
If you just look down the Q&A section.
Laura, will they be in the chat room or the Q&A?
They're all in the Q&A. So
okay.
Can encourage everyone to submit any questions in the Q&A. That'd be great.
Okay, all right, great.
Maybe I can ask you a question.
So, yeah, so go ahead, Bill.
So why is it that some NMDA antagonists cause psychosis and others don't? I don't quite understand what, and I know it's different subunits in the NMDA receptor, but what's the downstream difference?
The downstream difference between the NMDA receptors with different subunits?
Well, yes, exactly, yes.
Yes. So you get, so that there are differences in terms of regional distribution where some of the receptors that contain only certain NR2 subunits will be distributed differently in tissues. So for example, NR2B and containing NMDA receptors are expressed primarily in the forebrain and not as much in the cerebellum. By way of example, there are also differences in the electrical parameters, the duration of an action potential that's mediated, that they mediate, and also the current and voltage mediated during the stimulation. And so there are both differences in firing activity and in location. NR2B is also situated both extrasynaptically and intrasynaptically, whereas generally the other NMDA receptors are situated intrasynaptically instead. So there are those differences. I have some questions up now. Why don't I start with one interesting one? Ketamine has dissociative effects that require patients to remain in the clinic setting after administration, and esketamine subject to a REMS program, a risk mitigation program. And the question is psilocybin any different in this regard?
I cannot start my video. It seems to be, oh no, I can. Oh, great. Yeah, it's not any different. As I said, patients in psilocybin session are supported by a therapist throughout the entire session. It's a little more involved than supporting patients with esketamine or racemic ketamine. So there are special skills required and some interactions are required. But post-approval, I think psilocybin will be subject to REMS and similar level of, engagement as ketamine. So it's not going to be simply prescribed. You can't get it in Boots.
Okay, thank you. Mm-hmm. So I have a question for Richard Mead on what do you think the key lessons to be learned for research for psychiatric disorders from the conduct of studies in neurodegenerative conditions with such as with motor neuron disease?
I mean, I guess we kind of can learn. I think we can learn a lot from each other. I think the lessons are very similar in terms of. I mean, I must admit, I haven't really contemplated the unique issues, let's say, that you face in terms of kind of psychiatric drug development as opposed to kind of pure neurodegenerative targeted therapies. But I should imagine they are all pretty much the same in terms of being able really to understand whether you're having the intended biological effect in the target tissue. I think the problems you guys face are perhaps even more complex than the ones we face, given some of the complexities that we've seen today, all the way down to transcript level, all the way up to the kind of behavioural level that ensues from that. So, yeah, I think it's a similar problem.
Thank you, Richard. Here's a question for Liz. Liz, beyond CACNA A1C, are there other genetic targets that are emerging from psychiatric genetic treatments as treatment targets?
Yes, I think in a sense, almost the problem with genetic targets is an embarrassment of riches. We now have this vast array of things that are genome-wide significant, say for schizophrenia. And I think for me, the challenge is often narrowing that list down. I have some favorite follow-up genes. Obviously, you know, we're working on the whole VGCC family as well as CACNA1C, but there are certainly other genes that are genetic hits that we're beginning to work on. I guess, pragmatically speaking, I tend to take the approach of using rare variant evidence alongside evidence from GWAS studies to try and narrow down that list. I think if you can see, because GWAS don't give you genes so much as giving you bits of the genome, and we assume it's the nearby genes that mediate genetic effects, but you know, as we've seen in some of the talks today, that may or may not be true. I tend to use rare variant evidence as sort of a corroborator for common variant. And then beyond that, I think ultimately, I know there's lots of people using computational approaches to try and narrow down lists of genes, but I think there's nothing that substitutes for going away, reading the literature, doing your homework. So I have a postdoc at the moment whose job it is to kind of go through this massive amount of information, narrow down genetic targets, and then go away and research what we actually know about them. You know, are they druggable? What do we know about what they do in the brain? Because I think, you know, as we heard from John, what we need to do in the first instance from the genetic studies is to identify the low-hanging fruit genes that we have some evidence are involved in processes, say synaptic plasticity, that we know are important for psychiatric disorders, and focus in on them that way. I think ultimately there is no shortcut through that process of, you know, doing your homework as to what these genes actually do. I'm not going to say which my current favourite genes are because, you know, we have funding applications for them at the moment. But yes, we are beginning to move beyond the calcium channels. And broadly speaking, my way of doing that is to look for the intersection between common and rare variation. One thing that we were talking about in the panel discussion, sorry, the breakout room discussion earlier, which I think is an important thing to note, is that I don't think that it should be a prerequisite that a target has genetic support necessarily in order to be taken forward. I know in the US, for example, it's extremely difficult to get NIH funding for anything that isn't GWAS significant. And I think that's an extremely short-sighted approach. I think, you know, in many cases, the targets that we might want to focus on may not themselves be genome-wide significant, but they may be interacting with genetic factors that are genetic hits in some way. So I think we need to be fairly open-minded as a field about, you know, what makes a good target. And I think ultimately that will be, you know, it comes back to this kind of notion of working collaboratively with people with lots of different sets of expertise. You know, I can identify something from the literature as a neuroscientist that looks like a sensible thing to research, but I need to speak to people like John to know if there's any hope in hell of drugging it. You know, we need to take evidence from lots of different types of study and work together, really, I think, to make sense of all of this information.
Okay, and John Atack, if you could answer the question as well. Another questioner asked us, actually, I think it might be the same questioner, pointed out that since you're based at the MRC Psychiatric Genetics Group, the same question to you: what other genetically identified targets might be of particular interest in psychiatry?
So one of the reasons for moving to Cardiff was to link up with the guys at the psychiatric genetics group there. So Jeremy Hall, Mark Cohen, and all those guys. And we're working very closely with them to look at what all this information means. As Liz mentioned, we've got lots of information coming out of these genetic studies, but how do we look at all the pathways and identify the key nodes of where we can interact and what the proteins are at those key nodes, and then think about, well, are those proteins actually druggable or not? So we're working very closely with those guys to identify. It's largely a bioinformatics process, triaging through all those potential drug targets. And then it comes down to, as Liz said, reading the literature, knowing the targets, and trying to figure out if there is a way forward. So it's a huge effort. It's not trivial. These things don't just pop out and stand out. But there's a lot of legwork involved, not only identifying the targets, but also doing the target validation work. And so, as you've seen with the CACNA 1A project that Liz described, that looks like, oh, that's an interesting druggable target. But when you start digging into it, it's a bit of a nightmare to figure out what you would do from a pharmacological point of view. So there's a lot of heavy lifting involved between the identifying the gene and actually demonstrating it's a good drug target, and you can actually launch a formal drug discovery project off that. And a lot of the legwork is involved in overly expressing, underexpressing the protein in a cellular system that might have some disease relevance. And again, that's a complete other can of worms as to what's a disease in a dish for schizophrenia, for example. So it's not trivial. And I'm sure that, if it was trivial, everybody would be doing it. So there's not too many low-hanging fruit, unfortunately.
Thanks, John. I have another question for Ndaba. Have you considered, Ndaba, have you considered an informatics approach for identifying clinical candidates with a suitable profile? And could that broaden your approach to other disease areas?
Yeah, absolutely. It's a good question. I think informatics is a really sensible, systematic approach, and and this arena is expanding with various kind of computational methods and AI, etc. And I think this is a very good kind of starting point. I think this obviously needs to be married with other considerations as you sort of go along the process. So, having a broad base is good, and it gives you sort of creative ways in. I think once you the closer you get towards the sort of patient population, that starts to become a kind of bottleneck in terms of what's usable, not only from an efficacy point of view, because I can give you a lot of good data in terms of the efficacy, but also from a perspective of things like safety, for example, and quality of life, in conditions where you are already using other drugs or other treatment modalities like radiotherapy, for example. You have to sort of consider how other things kind of map into that whole process. So informatics is certainly a very good way, and we have been looking at that, and there's certainly been other groups even before us who've been looking at this quite extensively. I think it's important to then also look at other factors other than just efficacy when you look at whether that drug is usable or not in the real world. And this is where actually, again, other groups such as patient advocacy groups and charities are important, because you're not only looking about, can you extend survival times, for example, you're also looking at, well, how does that extended survival time impact on the patient's quality of life, for example, and how does that factor into the family and carers around that patient also? So, yeah.
Thank you. This is a question for Nicola. For translating mouse to human studies, are brains of individuals who suffered from Tourette syndrome donated or used to understand specific brain regions involved or the SNPs involved?
So we haven't used any Tourette's syndrome people donors at the moment. I think it's probably fair to say that there's some disease donors that are kind of more prevalent than others in the brain banks. I think we did a sort of a scan when we first started of what types of disease donors that we could get, which would obviously have a focus on our research. And I don't remember that. I mean, we did look for Tourette's, but I don't remember that Tourette's was particularly prevalent. I think it's kind of in some ways that when you're kind of looking for donor tissue, diseases where it affects families, they're quite motivated to donate, which is not quite so prevalent in Tourette's, of course, but you can certainly do it. And in actual fact, although we have focused on neurodegeneration to this point, of course, you could argue that the psychiatric disorders with more of a kind of a network effect would be particularly relevant to try for a technique such as NET-seq, where you're looking at specific neuronal circuits or something like that.
Okay, here's a question. Thank you, Nicola. Here's a question for Laura. How do we build on the increased drug discovery and clinical development in academia? And sorry, I just lost it. The Psychiatry Consortium funds early stage projects. What else do we need? And then a related question: and what can we learn from the mechanisms in dementia funding?
Yeah, it's a really good point. I think in setting up the Psychiatry Consortium, we've really learned quite quickly that there are an awful lot of gaps in the pipeline that need to be filled. We work in that early space of supporting academics to advance their novel targets and helping with those introductions and the support of industry into that drug discovery pipeline. But actually that's quite early on in the process, and there are actually quite a few roadblocks to development which we don't cover. And I think that's something that we're certainly discussing at the moment about potentially what is our offer for people who are later on in that drug discovery journey. So we've already identified with events like this and having discussions with groups of people that there are further hurdles a bit later down the line when you have a drug and you're developing it, and yet you still need that industry input and that industry expertise and know-how. So that may be more applicable to people that are working in SMEs rather than basic scientists. And I think that's definitely something that we need to look at and address. I think it's something that we should definitely be doing, and there are definitely plans in the pipeline for us to gather stakeholders that are all invested in the entire pipeline, the entire chain, and figure out where that support exists. So there is support out there available, such as other consortia who are doing this well. So if it's a case of directing people to those facilities and those resources, then that's great. But if there isn't anything there, then we need to be discussing with funders and investors and all of those types of people as well, to see where their interest lies and how we can get everyone together to address this problem.
Okay, thank you, Laura. I'll take one now for me. Is intranasal esketamine associated with any cognitive impairment? And then a related question was, are there long-term negative sequelae of chronic use of esketamine? The reason these questions were related is because that one of the questions is would there be not only what is the cognitive impairment acutely, but also would there be long-term effects on cognitive impairment? And so in our studies, we addressed that in a couple of ways. One was with respect to acute cognitive impairment. We characterized the effects in healthy individuals and that with repeated cognitive testing. And what we learned was that you did get prominent effects on cognitive function at 40 minutes post the start of treatment. And those were by and large back to normal by 2 hours with very few exceptions. And the most potent effect was on the Groton Maze Learning Effect test, which was so potent that we wondered whether that could be a useful pharmacodynamic tool. Interestingly, patients then were, or controls were tested repeatedly, and what we found was that you ended up the individuals who, and so these again were healthy individuals, the individuals who had received esketamine as opposed to placebo ended up showing better retest learning effects than the healthy individuals that were only treated with placebo. So we noted that was not a primary outcome, but it was just noted as part of the study. But it would go along with the concept that you're getting a long-term potentiation during the study. In the long-term study that I showed you, we were testing cognition at about 4-week intervals. And what we were able to show is that you also had improvement over time. You had improvement in cognition in the short term over a month, where we had an actual control group that was getting conventional antidepressant but not esketamine. There you had a slight advantage to the esketamine group over the oral antidepressant plus placebo group. And then over time, you had continued improvement in cognition, especially verbal learning, verbal memory and learning. But we did not have a control group that was studied over the course of that year. Now, FDA had the same concern about what about long-term use, and so they actually required us to extend the studies. That we had a study where patients that had finished our protocols could go to get continued treatment, and FDA required us to continue that out a total of 3 years. So we've had many patients now that have completed the 3-year, but we're still finishing up the other 3-year group, and then the rest of the 3-year group. And there we're also getting repeated cognitive testing across time. So I don't know what the results will be at the end of the 3-year period, but at the end of the 1-year period, there was no evidence of cognitive impairment. Let me go back to a question about psilocybin. There were several. There are super questions here. Do we understand in the clinical use of psilocybin what is needed for optimum treatment with respect to the depth of experience and duration of experience? Are they interchangeable, so shorter or deeper is the same as longer, more shallow? Do you deliver different benefits?
Yeah, it's an excellent question. The quality of the experience seems to matter. So the 2 studies that I mentioned, I think, looked at the quality of the experience as measured by the scale called 5 dimensions of altered states of consciousness that characterizes kind of retrospectively the quality of the experience. And the deeper the experience, the more of ego dissolution in the experience, the better the outcomes or the longevity of the effects seem to be. However, the studies are small and underpowered. Those are just early signals, could completely disappear in larger studies. There is a great deal of interpersonal and intrapersonal variability. So the same person can take the same dose on 2 different occasions, will have completely different experience. So it's difficult to say right now, but it seems that when patients achieve that ego dissolution, that kind of peak experience, they will have better clinical outcomes. We're going to continue looking at it, and that's something that we're looking at in phase 2 studies. So we'll keep an eye on that.
Okay, great. Thank you. Mm-hmm. So I'm mindful of time. Bill, I believe I'm supposed to turn it over to you for wrap up and closed session at this point. Bill, are you there?
was unaware of that commitment. Was it on the bottom of my instructions to the?
It's at least on the schedule, you know. Unless Laura wants to wrap up.
Okay, I just wasn't aware of that. Well, how are we doing for questions? I mean, I thought we were going to extend the questions a little bit further. Is that? Are we?
Yeah, I think we've got quite a lot of questions not answered
Okay.
yet. So if
You know,
if
we do have a lot. We have a lot of
then
questions. Yeah. Yeah.
we can maybe
Okay.
do another 10 minutes on questions if you're happy.
Okay, Laura. So I can go another 10 minutes then.
That'd be great.
Okay. Okay. All right. There's a question. Another question that multiple or that also was addressed to me. Can esketamine induce manic symptoms? We were concerned about this possibility as well, and so we excluded patients with bipolar disorder from our studies. There have been 2 academic studies that looked at bipolar depression and showed it reported efficacy, but it has been generally understudied. We only at the time that we had submitted our data package to health authorities, we had studied more than about 1,800 patients, and we had 2 individuals who, although they were admitted into the study as unipolar depressives, they had in fact had one had manic symptoms and the other hypomanic symptoms, and so we by protocol we dropped both of them from the study. Interestingly, they both felt that they had benefited from the esketamine. So one of them went into a ketamine clinic outside and continued to receive ketamine. So the rate that we encountered precipitating mania or hypomania among a group identified as having unipolar depression was low relative to what you'd expect for antidepressant pharmacotherapy in general. But it was not zero. Let's see. Maybe I could ask Bill. Bill, maybe I could ask you a question. What are the mechanisms of esketamine and psilocybin informing us about the neurobiology of mood disorders? There you go.
Thanks, Wayne. I really appreciate that question. I think the common factor has to be glutamate, doesn't it? Because 2A receptors modulate cortical glutamate neurons and how active they are in releasing glutamate. So there could be some commonality there. But one PNAS paper that always bugs me, I'm always talking to Joe Neal about this paper, that psilocybin has quite potent affinity for tumor necrosis factor. I mean, could that conceivably be relevant? And I know also that some cytokine mechanisms, I think you need IL-6, if I remember rightly, in order for ketamine to have its behavioral effects. So what do you think? I mean, do you have a theory? I haven't heard of the theory. Maybe Von Weider does, but if the 2 work in the same way, I'm burbling, and I don't know how you would test it out, really. Yeah, glutamate seems to be a key focus, particularly presynaptic glutamate function that 2A receptors modulate, as well as the NMDA ones, and they both increase presynaptic glutamate activity.
Okay.
So all the downstream things you mentioned about what the consequences of AMPA signaling apply to both.
Yeah, that's a good.
I think a lot about schizophrenia these days. I have been interested in ketamine, but I'm not up to the minute with it. What do you think? I mean, is that right? I mean, they both do powerfully disinhibit glutamate release, right?
You know, I think you came up with an excellent question. I mean, an excellent answer. Yeah, yeah. Okay, Katerina, do you have anything that you would add?
No, I think I didn't think along this line. So yeah, I think it's a good hypothesis.
Okay.
I like the idea that both of them might interrupt rumination. You know, I think that seems particularly compelling with psilocybin. That what keeps depression going and chronic is this constant sort of ruminative thing that you can't break the cycle of, and that by disrupting the default network, maybe both drugs can just interrupt that and show that it's possible not to be ruminating and thinking negatively the whole time. So I do wonder how much you need the psychotherapy and how you would do the clinical study to show that, and also the dosing. I mean, low doses of ketamine better than high. How far down can you go with psilocybin? There was a question from the audience about low dose psilocybin, microdosing.
Yes.
Yeah, microdosing is different from low dose of psilocybin. Right now, so there is no evidence that microdosing is either safe or effective for treatment of depression. And by saying that, I really mean that there is no evidence. Maybe somebody could go and do the studies, a microdosing, high quality study, to generate some evidence, but it's very technically challenging, logistically challenging. And also physiologically, it's hard to imagine that psilocybin would be a good candidate for microdosing. Because there is an evidence for LSD, and again, mechanistically it's possible and plausible, but not for psilocybin. I've had extensive discussions with two researchers looking at it, Dave Nichols and his son Charles Nichols, and all the work that they've done, goes against the notion that psilocybin microdosing is effective for anything. It just simply doesn't hang around receptors long enough, or is not strong enough, and doesn't trigger the same cascades. In terms of low dose of psilocybin, low medium dose, again, the selection of high dose is based on the fact that this particular type of experience is dose dependent. The higher the dose, the more chances that people will reach that sort of critical mass of downregulation of default mode network, and will have that experience of dissolution, for the lack of a better term. And it seems to be dose dependent. So the higher the dose, the higher the chances that people will achieve clinical results. It's also, in a way, it's counterintuitive, but it seems to be safer to have a higher dose because the lower doses leave room for negotiation for a lot of anxiety. So people try to argue with this unusual experience without onset of unusual experience. Whereas when the dose is higher, they just go under with much less struggle, and then stay in this experience for a longer period of time.
Okay, thank you, Katerina. The next question, although it could go for anybody on the panel to comment on, but I wanted to hear Liz's answer to this one. So does greater emphasis need to be placed on stratification of phenotypes of psychiatric disease? And I would add the question about genotype in addition to phenotype, to further refine or the development of effective treatments. How can this be done? And I wondered, in your case, Liz, would you pick, if you were going to stratify by genotype, do you think you'd have to pick the specific, like CACNA A1C, for example, or could you also pick people who had CACNA A1B and D receptor mutation, because those have also been identified in bipolar and unipolar patients?
Yeah, I think it's a really interesting question. I mean, as many of you will know, the idea of refining the current diagnostic criteria is not a new one. It's something that many people, including many of the guys in Cardiff, have been working on for a really long time. I think at a phenotype level, the challenge is knowing where to go with that, because Maiko in particular spent years trying to refine phenotype and then look at the genetics and see how the two map onto each other, and it's just proved incredibly challenging. I do wonder whether, as we start to understand more about how genetics maps onto brain function, so things like the Enigma project and the biobank imaging data, where they're collecting imaging data from large amounts of people, and they have genetic data as well. If we can kind of refine almost kind of brain phenotypes rather than sort of clinical phenotypes, it will be interesting to see if that helps or not. I think either way is an interesting question. I suppose the genetics, in terms of the clinical phenotype, are beginning to tell us which disorders overlap more with each other. And I think for me, it perhaps wasn't as it wasn't what you guess in a way. I suppose if you'd asked me genetically which disorders I would have thought go together most of all, I mean, obviously, I think historically you'd say maybe bipolar disorder and schizophrenia because there's a lot of overlap, but actually, you know, there's a lot of overlap between schizophrenia and autism, and I think that for me was kind of surprising, although in retrospect, both of these are probably conditions that have a large neurodevelopmental component. So, you know, I think the genetics in terms of the clinical phenotype is perhaps for me most useful in clarifying ideas about what might be the underlying brain mechanism, you know, development rather than things happening later on. I guess in terms of stratification for clinical trials, I think again it's a very nascent question. I guess with CATNA1C, in terms of the rare variants, there are some rare conditions like Timothy syndrome, which many people will be familiar with, which are caused by mutations in the CATNA1C gene that lead to a clear gain of function. Now, those are conditions where you could easily stratify, you know, you clearly want to reduce channel function in that particular condition. The challenge, I think, with regards to the common variations, we still haven't quite fathomed what it is that it's doing to channel function. You know, my working hypothesis is that it's something to do with splicing, and we're looking into that at the moment, but it's one of the genes where there isn't a clear link between genotype and phenotype, even at the molecular level. And then I think if you haven't got that, extrapolating up to the level of the whole individual becomes incredibly difficult. I think for me, a sort of pragmatic thing would be if you have a, say we develop a calcium channel blocker, I can see that it might be useful to at least understand how genetic variation across all of the calcium channels influences response to that blocker. But at the moment, I don't think we have enough information to a priori say, yes, you should stratify on the basis of this particular genetic variation outside of the very rare variants. But I think studies that aim to understand what genotype is doing to the molecular kind of milieu of the brain, and then pragmatically relate that to brain function as an intermediary, is probably the best hope we have. I mean, I would hope that we'll get to personalized medicine, but at least in terms of genetics, I still think, apart from the rare variant studies where there's really clear molecular effects, we're way off that at the moment. But that is very much with a genetics point of view hat on, so it'll be interesting to see what the other panelists think.
Mm-hmm. Oh, okay, thank you, Liz. Laura has allowed me one final question, and I think I'll take this one because there's a very interesting question about pointing out that CP101606 or Atrexprodal, which is again the NR2B antagonist, does not select for open channels. So how does that tally with targeting rapidly opening and closing channel on GABAergic idea? And then two related questions: memantine, why is this not antidepressant? And then finally, the dopaminergic effect seems more reasonable for induction of effect, but still does not explain selectivity. So the CP1016, I mean, this is a correct a statement about a difference between CP101606, which is not an open channel blocker. Nevertheless, the NR2B antagonists were tested by the Yale group and shown to have the same increase in glutamate release. In Jerry Sanacora's group did this with magnetic spectroscopy, and then Ron Duman's group did this preclinically and showing the same mode of action of increasing glutamate transmission and synaptic plasticity around the glutamatergic synapse. But it's a good question because there are differences between how an allosteric compound, which the NR2B antagonists are, may differentially have effect. There are some questions, for example, about whether the CP101606 worked as quickly. In Sheldon Prescorn's study, it was effective by 96 hours after, but not 1 day after. So there are some questions about differences in the clinical characteristics of the antidepressant effect. Memantine, we think, was simply not tested. It was tested in depression, not effective. and we think it just wasn't used in the high enough dose range to get NMDA receptor antagonist effects that would trigger this cascade of events. And then the last question about the selectivity, I think, is also useful, and this relates to some of the other questions. I received a couple of other questions about a study that identified naltrexone, the mu and kappa opioid receptor antagonist, as reducing ketamine's effects in a Stanford study in which they ended up stopping the study early because they got severe adverse events by combining the two together. And so it was a study that didn't have appropriate controls to look at the effects on placebo or to look at other antidepressants. But in general, non-selective effects involving, so even though ketamine, esketamine are about 2 orders of magnitude too low in concentration to have any mu-opiate receptor effect directly, and we didn't see any mu-opiate receptor adverse events like respiratory depression, for example, or pupil change. But nevertheless, all antidepressants are thought, and placebo effects are thought to depend on endogenous opioid transmitters, and so those endogenous opioid transmitters work in synergy with the dopaminergic mechanisms to give a normal happy mood, to give the positive expectancy following placebo, and so there is a role for opioid peptide transmitters internally, also in the antidepressant effect. All conventional antidepressants have been shown to raise opioid peptide release, and so it may be something that's more general that still deserves mention. I guess the one caveat I would say is there have already also been 2 studies that are about the same small size that found no effect of naltrexone on the antidepressant effect of ketamine or esketamine, and we also did not find any effect on the ketamine esketamine effect or esketamine effect with mu-opioid gene variants that have been associated to affect responses to opioid analgesics. But interestingly, the opioid gene variants did affect the response to the placebo plus oral antidepressant group. So it shows that the importance of considering those indirect effects is certainly relevant. But at least ketamine, esketamine at antidepressant doses don't have any direct effects on the opioid. So with that, this has been a very interesting set of questions. I appreciate the audience participation and thoughtfulness, and also I appreciate the panelists coming up with such thoughtful answers to the questions.